Good day. In this lecture, we will be discussing about the different techniques in molecular biology. So in this particular subfield, so we have the genomics field, the proteomics, transcriptomics, and metabolomics. So first off, let us look at how the different fields relate to one another. So going back to your lecture in molecular biology, so this illustrates the flow of the information encoded in your DNA and how it is being expressed. Now, first we have your genes, of course. The starting point is, of course, the source of the information, which is the genes in the DNA. So the gene sequences studies of that, this we call this one genomics, the study of the genes in the cell. And then we have these genes are being transcribed to RNAs and they are being expressed when they are transcribed. So we have your transcriptomics. So genes versus transcriptomics, there are different applications on these different fields. For the genomic side or the genes, looking at the genes only, we would know what available informations there are. But in the transcriptomic side, we will see that which of the informations are being used or active informations, because some of the informations are dormant. And then we have from the transcriptomic side, we have the proteomic side. Now proteomics are also concerned with what are being expressed, of course, what is existing already. Now there is a I guess it's called an issue. There's a thing between transcriptomics and proteomics is that some people prefer to get proteomic data as opposed to the transcriptomic data because it provides a more clear view of what is being made, because of course the products of the genes are the proteins. And the transcriptome, the RNA, is just basically the go-between between the two. And we have post-transcriptional modifications that might occur that we have this transcription, but there are different ways to translate or to make the transcribed information into different proteins. We can have one transcription to multiple types of proteins because we have alternative splicing events that might not be seen in the transcriptomic data. Now that is in the proteomic side. We will see, of course, that one in the proteomic side. Now, the issue with this proteomic side is that it's very difficult to do. It's a challenge to know, although recent innovations, especially in the mass spec, have made large leaps into knowing the proteomics, but still there are some there are things that need to be done or improved on the proteomic side with regards to the actual laboratory setting or the actual implementation of the method in the lab. And then the last one is the metabolites, the metabolomic side. So now this is because proteins and not exactly most, proteins are also enzymes, and the metabolomics looks at the products of these proteins. So it's more like if the proteomics is the functional view of transcriptomic, then metabolomics is the functional view of proteomics. Why is that? So because even if the protein is existing, which we can see in the proteomic side, sometimes it's inactive. So it's there, it's existing, but it's not doing anything. So we can look at the metabolomic side to know that this is the actual products of the proteins, the movement of the enzyme. So we have an actual product. But I'm not saying that metabolomics is the ultimate real view of what the cell is, what really is going on in the cell, but rather we are looking at the actual product. It's like looking at the factory. So in the factory, you have or in a plant, a factory or manufacturing plant, you have the plans or the plan, or how should I say, the SOP guidelines in the DNA. And then the transcriptomic side, you have the bosses, or the, how should I say, the plant managers, the managers and the supervisors doing telling doing the planning or telling the workers to make this type of product, this variant of the product, do this and do that. So they are somewhat like the implementation side, but the actual workers are the proteins. Now that's the proteomic side. And then we have the metabolomics, which is the actual product output. So let's say, for example, in a bottling company for a soft drinks, So the genes, we have this SOP plan. These are usually the file folders for how to make the product. And then you have your plant manager in the transcriptomic side to tell which product would be manufactured by that day. So you have the managers going down to the supervisors and so forth. So that's your transcriptomics, the messages. And then we have the proteomics, which are the actual workers, the rank and file workers that actually do operate the machinery and do the to manufacture this product. And finally, we have the product, which can be analogous to the metabolomic side. The how much of this soda soft drinks in can was produced, how much of the soft drink in that particular flavor was produced, how much of that was produced. So that's your basically metabolomic side. So all in all, not one thing can represent the whole. So all of them, we need to have information from all of them to know the actual what is really going on inside. And depending on what your question is, what's your research question, what you want to know, you would use either the genomics, the transcriptomic, proteomic, or metabolomic data. So anyway, that's your that's a thing in that's how you do studies and research in molecular biology generally. Okay. Now, genomics, as you can see, this is about the genes, the genome, genes, the DNA. So how do we study the DNA? So the most common right now is the PCR, which is quite a revolutionary technique for your PCR. So rather, it has been to the forefront now because of the pandemic. So previously, only the us basically the people who had been into molecular biology, only we are the only ones previously that know, or not exactly only, but it's not really a familiar term for the layman before. But with the pandemic, because the only sure way to know the presence if you have COVID is through the swab test, and implementing the swab test is through the use of a PCR. It has become quite a common word for the many people right now. Anyway, so how does it actually works? So in PCR, it's polymerase chain reaction. It's basically like a Xerox machine, but you are photocopying the DNA instead of papers. It's a Xerox machine for DNA. So how does it work? So first you have your templates. So this is your genes that you want to use. Now take note, you cannot have a whole chromosome into a PCR. You need to cut them up into manageable bite-sized pieces. So a PCR can only amplify fragments of the DNA, not the whole genome molecule. Okay, so it must be a manageable fragment. Anyway, so you have your strands. So because the DNA is double-stranded, you have the two template strands. And this is if you look at PCR in our laboratory, you would see it's called thermal cycler. So some company did have this thermal cycler because it cycles temperature. So how do I mean by cycling temperature? So it goes from one temperature to another in a cycle. So the first step is, of course, denaturations. So denaturation, the PCR will increase the temperature to 95 degrees or sometimes higher. Actually, it depends on the GC content of the strand. So the higher GC content, because the G and the C nucleotides have 3 hydrogen bonds, it's more difficult to break up. So the greater the GC content, the higher the temperature. So you have this, but the usual SOP protocol is 95 degrees. So you have this 95 degrees. This is the denaturation phase. And then you go on to your second step. So once you really won't know, or rather the most thermal cyclers do not have because actually you can see whether you have denaturation in a microscopic equipment, but most thermal cyclers are not equipped with that function. Anyway, they just give it for a few minutes to a few seconds. And once it has been denatured, it goes now to the annealing step. So the temperature goes down to the annealing temperature. Now, the annealing temperature depends on the primer. And the primer would have a certain GC content. And the GC content of the primer is the one that would dictate the annealing temperature. But it's around this temperature, 55. So it's around 50 to 57, 56, even a bit higher. So depending on the GC content. So primers, the primers are the one that would jumpstart the reaction. Because remember, in DNA replication, you have your the primers are actually done by the mRNA polymerase, the RNA polymerase, and then followed by the DNA polymerase. Because the DNA polymerase cannot start it cannot start an elongation without a primer. There must have been already a strand of DNA in there where they would just add the nucleotides in. The polymerase cannot add from nothing. They cannot add nucleotides without a primer. So but instead of just having an RNA polymeraser, we have a primer. And this primer is actually what makes PCR selective, because if you have a certain primer that is selective for a certain gene, only DNA with that template strand would be amplified. That is actually one of the principles of your the swab test, the the PCR test for COVID. So if you have the DNA for COVID or a COVID DNA in your sample, you have a primer selective only for COVID DNA. So if you have many other DNA there, only the DNA from the virus would be amplified and no other. And that's because of these primers. Now we have two types of primers, the forward and the reverse primers, because you have two strands of DNA. So remember, double-stranded, they are anti-parallel, so 5' to 3', and the other is 3' to 5'. And then you have your polymerase. So after annealing to a certain temperature, you would now ramp up the temperature again, increase the temperature to 72. The 72 degrees temperature is actually the optimum temperature for the polymerase, not our polymerase. We are using a polymerase. Before, it was a polymerase extracted from a certain bacteria that was living in a hot spring. So it's a thermal aquaticus. That's why it's called TAC. TAC polymerase. That is the name of the Thermus aquaticus, polymerase from the Thermus aquaticus. That's a TAC polymerase. That was the conventional one that was used, but current versions of the PCR have a more robust version of this TAC polymerase. Anyway, in the synthesis stage, you ramp up the temperature to 72, or to what the optimum temperature of the polymerase in the kit is, so that the polymerase can do its job. So this is usually the longest part of the cycle. So you have you need to allot a few minutes for this one, for the amplification. But this is actually quite fast. They add nucleotides in a fast manner. Now, once you have a new strand that once the polymerase have a new strand, you would then repeat the process. So you go back to 95, and then to 55, and then back to 72, and then repeat the process again. So you would do this for several times, around 20 to 40 cycles. So that's why it's called the thermal cycler. So this is how PCR works. So you do this until you have amplified your DNA in such an amount that you can actually visualize your strands, or rather the presence of these strands in your agarose gel electrophoresis. And that is basically your PCR. Okay, now moving on, the next step after this, how do you know if you have amplified the correct strand, or you have these necessary fragments amplified in your DNA? So the conventional method is to use agarose electrophoresis. But just a quick note, qPCR right now, we can do that in real time. We can know if there is an amplification going on real time through qPCR. But what I have shown you is the conventional PCR. And that's conventional. So the conventional PCR, in order to evaluate the results of that, you need to do electrophoresis. So here is electrophoresis. So you have a gel electrophoresis because it's in a gel. You are using a gel as your medium, and it's submerged in a buffer. So this is it has, of course, this is a buffer with ions, and it's electrophoresis because you have electrical charges going around in your sample. So your gel is submerged in a buffer solution, and there are electrical currents going on here. So you have an electrodes here, a negative and a positive electrode. Now the DNA fragment is negatively charged because of the phosphate groups. So you have the tendency of the fragments to migrate towards the positive electrode. And therefore, you need to have a proper positioning of the gel. So the gel is, you have this gel. In the gel, you have a well in the gel where you would put your samples. And of course, when you run the machine, the equipment, of course, there's a live electrical current going around here. The fragment will migrate towards a passively charged electrode. So you have a migration here. Shorter fragments go first or go faster than the longer ones. So you would see here bands in the gel. So the gel matrix, they can be polyacrylamide or agarose. So the difference is for polyacrylamide, it has high resolving power. So if you have DNA fragments of very similar lengths, but you want to separate them out, you use polyacrylamides because they have higher resolution. But agarose is the generic one. You usually see them in the lab because it's what's cheaper, it's easier to use than polyacrylamide. And although it has less resolving power, but it can still separate DNA molecules in terms of tens to hundreds or thousands of bases. Because for polyacrylamides, you must have a very short DNA fragment for that. So that is why for the gel, we usually see the agarose because generally the resolution is enough for the agarose. We don't really need the higher resolution of the polyacrylamide because the differences in the genes or rather in the DNA fragments is usually up to several hundreds to thousands of bases difference is the usual requirement, not 10 bases. And also, if you want to know the differences between in the DNA fragments of a certain, say, 5 bases only, rather than do electrophoresis, why you just need to do sequencing. It's much faster and easier to do that and more accurate to do that rather than do electrophoresis for resolving that size range. So you have, that is why polyacrylamide is not usually seen in the laboratory for the DNA. We usually have agarose. Okay, now here is a different technique for the electrophoresis. So if you have very long DNA molecules and you want to resolve them in a more you want to improve your resolution in an agarose gel, you do the pulse field gel electrophoresis. It's a slightly different equipment for your than the standard electrophoresis setup because as you can see, you have 4 electrodes here, and you have pulses here. So the movement is a bit of a diagonal, and so it's just like shaking up the DNA a bit so that you would have a higher resolution for the larger fragment. So that's the pulse field electrophoresis. So you have alternating pulses of electric charge on the left side, right side, left side, right side. Okay, now we have this denaturing gel electrophoresis. So another variant of your electrophoresis. So usually that's the DGGE. So this is usually seen in microbial samples. So you have samples from different microbial communities, and you don't want to, or rather, it's difficult to separate each species from one another. So you have a mixture of different DNA from different samples here. So you will run them to a PCR. You are adding a GC clamp, which anchors denatured DNA in your fragment. So what happens is that you add an additional dimension in your electrophoresis setup in such that when you have denaturing gel electrophoresis, when your when your fragments or PCR, your gene fragments are denatured, there is you have this movement, so you can separate out your unique genes in the communities. So how does that works? Basically, you are immobilizing the genes that are common by clamping them. And if you do not have a clamp for that, what do you mean by a clamp? So it's basically because the genes hybridize one another. So single-stranded genes would go forward, and the shared genes, the double-stranded ones will be left here, while the genes that are denatured, they do not have a hybrid because you don't know them. They are unique ones. It is in forward. So basically, that's DGG. Now, another one to know, or rather to use in PCR and also in electrophoresis, is the use of probes. So probes are, it's now ubiquitous in molecular biology, especially for the genomic application. These are DNA, short DNA strands, fragments, where it is the complement of a certain part of the DNA you are looking for. So you want to know, or you want to know the presence of a certain fragment, you need to have a probe for that. So it identifies the gene you are looking for. So it identifies by hybridizing, because the probe is actually another DNA fragment that is specific, that is complementary to the specific gene fragment that you want. So it's you have your probe. So you would hybridize it to your fragment. So it's actually one of the um, core of Southern blots. But anyway, so here is the use of the fragment. So these are the probes. So if you have a DNA, you denature it first. Then you have several probes. So this serves as a beacon. So these probes have an attached fluorophore. Now it's fluorophore. Before it was radioactive, uh, a radioactive material that they attached, but because of the danger of the use of radioactive substances, you have now fluorophores. So so that you can see where the probe are attached, and you would be you would be able to identify the presence of a certain DNA there. So, and this is where probes come in. So we have the Southern blot hybridization. So for the Southern blot hybridization, it's actually very similar to a certain PCR. Basically, you extract in here, this is actually your DNA. So you extract DNA fragments from your DNA fragments from your sample. You cut them up with restriction enzymes. You get your fragments now, and then you run them on electrophoresis. So in here, you don't have to do PCR because of the abundance of the DNA from the sample. So you just need to cut them up into fragments and then run them into PCR. Now, you now have a mixture of different fragments of the whole genome of the chromosomes of your sample, but you want to find specific ones. And that is why what you would do is transfer the genes, the molecules, to a nitrocellulose gel. So you have your, this is your gel from the gel electrophoresis. You would put your a nitrocellulose gel here, and then have a filter paper above. So it takes a few hours, and so that the molecules, the DNA, will be transferred from the gel to the nitrocellulose membrane. And then the nitrocellulose membrane will be incubated. It will be submerged in a solution with the labeled probe. So the labeled probe will attach to the gene fragments where it is complementary to, and then you would be able to see. Now, this is autoradiography because before they're using radioactive probes, but now we can have fluorophores instead of radioactive substances. So it's under a fluorescent. It will fluoresce. Now here is, for example, you have this ladder of DNA. You want to know a specific site where the gene you want is. So by autoradiography, it would select out only the line where you have your DNA of interest is. So basically, that's Southern blot hybridization. It's electrophoresis with an additional step. So another view of your electrophoresis step. So the additional step here is the transferring to the filter and the use of radioactive or rather labeled probes. Now we have RFLP analysis with your southern blot. So this is restriction fragment length polymorphisms with an additional step, which is the southern blot. So this is how you actually identify paternity. This is usually done in paternity test and even in forensic test, because in forensic test you have 13, if I remember correctly, it's 13 labels, 13 probes for identification of individuals. And that 13 probes would be hybridized in a or into your nitrocellulose gel fragment you are using southern blot. The operation here is basically southern blot with, instead of using one probe, you're using 13 probes. And so this is why you have your ladder. You will end up with a ladder because you have 13 probes. Now, it doesn't necessarily mean that you would have all 13. Sometimes you have, sometimes you don't have, but each person has a unique pattern and length. So that probe can attach to a certain length of a different size. So it would fluoresce a certain color, but it would have a different size. So the thing here is that you have now several lines of, it's like a barcode. You have several lines of several spaces between each, which is unique to the individual. And that is actually how you do identify a certain individual from that. Now, for paternity testing is that you would be able to know if a person is related to another because they would share common bonds. So for example, here you have the mother and the father, and these are the children. So you would see daughter one, daughter two, and sibling one or son one have they have samples, or they have these fragments from their parents, especially daughter one and son one. So you would see that daughter one has a band that's of the same length as the mother, and another with the dad, and here is the dad and then the mother. So that it doesn't necessarily mean that all of the bands from the dad must be seen on the daughter. Doesn't necessarily have that. It's just that all of the bands that is in the children must be seen on either of the parents. Now, if there's a band that is not seen on either one of the parent, that person is not related to that to a parent, or there is something else going on here. So for example, here's daughter two. So you have here a band consistent with the mother, but it's not consistent with the father. So that means this person, daughter two, is the child from a different father. And then we have this one doesn't share much of a bonds from either parent, so this one is adopted. So basically that's how you do paternity testing in using DNA paternity test. Okay, now southern blot has been one of the very how should I say this, it has become popular in molecular biology labs, especially in the previous decades. And they apply this technique into different areas of molecular biology, like RNA and proteins. In the proteins especially, it has become popularly known as the Western blot. Although Southern blot, it's named Southern not because of its geographical location, but it is because the one who designed the technique is actually named Southern. His name is Southern. Surname, I mean, is Southern. And so they call it the Southern blot. But of course, consequently, or rather succeeding scientists and researchers, when they use the technique, we're using southern blot. And then we want to apply to an RNA. So it's a variation of the southern blot. So they want to have a different name, but they want to keep because southern can refer to a geographic region. I don't know if it's a deliberate change to use geographic locations, or it's just a mistake that they thought it's called southern because it's from the southern area or south states because it's in the US. And so they have the northern blot, which is for RNA. This is southern blot but applied to RNA. And then we have the western blot, which is again another variation of the southern blot, but the application is on proteins. So that's how those names came about. Anyway, so for the northern blot, the same issue you have, you would transfer, you would transfer it in an electrophoresis. You transfer the fragments in a gel, or rather in a filter or a gel, a nitrocellulose membrane filter. Well, basically it's nitrocellulose. It can hold your DNA without allowing it to move, unlike the gel. That's why we need to transfer it. When in gel, it might move around still, but in the filter, it's fixed in that location. Anyway, and then you incubate it with a probe, you add a probe there, and you would see highlighted regions where the probe is at. So basically that's blotting. That's your southern blot. And this is for the northern blot, the same technique, but using RNA. And then another variation, but not exactly in the blot. This one is fluorescence in situ hybridization, the use of probes again, but this time it's a fluorescent probe. So you have here your probes, colored probes that you incubate into a cell. These probes may be DNA probes for specific DNA. It can also be protein probes. So these probes can bind to the specific targets and it will give off a certain color. And when you apply it in a culture of cells or whole cells fixed in a microscope slide, and you apply the probes there, it would of course stick to the one that you wanted it to stick, and it will fluoresce. And when you view it in a fluorescence microscope, you would see the light regions, the regions where the probe is at, light up. And that is fluorescence in situ. It's called in situ because it means on site. It attaches to where it is originally on the cell, as opposed to the southern and northern blots where you have your the gel is already run in the gel. It's no longer where it was at before when it was still inside the cell. In here, you will see where it's located inside the cell. So in situ hybridization is not, this is called FISH. It is not restricted to DNA. It can also be used in proteins. So here is for chromosome mapping. So you want to know in which particular part of the chromosome your particular gene is. So you would have a probe and put it in a slide with the chromosomes. This one is where the chromosomes have already been extracted, and it attaches to the regions where the probe is at. So here is another view of that. Of course, the region that lights up, these are where your probe is at. Okay, so that's it for the genomics approach. So I'll stop the video here. The next video, we'll look at transcriptomics.